am I audible to all of you? Hi people, am I audible to all of you? Yes? Hi everyone, am I audible? Hi. So I believe I'm perfectly audible, everybody. So welcome to the session, guys. Welcome to Unacademy NEET English. I'm your biology educator, Ambika Sharma. And of course, it is the part 3rd of the chapter cell. Right? So hope you guys are making the notes. Hope you guys are liking the sessions. And I hope you are practicing the MCQs as well. Okay? So let's start the class, guys. Let's start the class. So today we are going to continue the prokaryotic cell. But before that, let me tell you. So if you want to be the part of our batch in which from tomorrow, from day after tomorrow, I'll be starting a new chapter. So students, I am going to start anatomy and then plant physiology, right? I have seen this thing that students, what they do, like, of course, human physiology, they find it easy. They find it very interesting. They start with that, right? They complete it. But when it comes to plant physiology, students, they really don't appreciate it much. They find it very difficult. They find it very difficult to remember the things or to, solve the questions. But here in my sessions, trust me on that, you people are going to love plant physiology, right? So that is why in the starting of the batch only, I will be targeting the plant physiology first, and then we will go for the human physiology, right? Because I want to make sure that whatever topics are difficult for you, you really should find them easy. You really should know how to approach such topics, right? I'm going to simplify your NCRT. Along with that, I'll provide you the content which you need for your NEET examination. So keeping NEET 2025 level in our minds, accordingly, we will have the session. So guys, from day after tomorrow, that is from 1st of August, I will be starting anatomy and then plant physiology. Because right, if you will not join, right, if you will not study the what do we call it, anatomy, the plant physiology will be difficult for you, right? Okay, done. So let's continue the session. Right, let's continue the session. So far we have completed the cell unit. Okay, so accordingly you'll get the revision sessions, whatever chapters like we have already completed. The live sessions will be there for them, but from the 15th of August, right? From the 15th of August. So I'll be taking the live sessions for that as well. So before that, please make sure you join the batch. So let's continue. Let's start the batch. Okay, let's start the batch. Okay. Okay, so this is what we have already discussed, right? We are done with the history part. We are done with the basic introduction part. We are done with the overview of cell. So if you people are having any doubt anywhere, you can freely ask me. And after that, we started the prokaryotic cell. So in the last class, we discussed about the overview of cell and the prokaryotic cell. And the last topic that we completed, it was beta gram negative and positive bacteria. So quickly in the chat section, students, do let me know that how do we differentiate gram negative and gram positive bacteria? Actually, what is the criteria for that? What is the criteria for that? Quickly tell me. Everybody, quickly tell me. Yes? Quickly tell me. People, let's make the session interactive. It should not be like that a teacher is speaking, a teacher is standing in front of camera and just speaking. No, right? Let's make it two-way interaction, which is not possible on YouTube, of course, but still, let's try to make it, right? Let's try to make it. So quickly answer my question. How do we differentiate that? How do we differentiate that? Gram negative and positive. Yes, what is the main point there? What is the criteria there? People, quick. Yes. So what is the criteria of differentiating gram negative and gram positive bacteria? Gram negative and gram positive bacteria. How do we differentiate that? You know that the main difference is of what? The main difference is of cell wall. And we discuss the procedure in detail, where we discuss one trick, that is come in and stain. Right? So I told you there are the steps here, like crystal violet stain. Right, you are going to stain with the crystal violet, you are going to use the crystal violet stain. Then iodine, then alcohol or acetone, then safranin. Right, then safranin. These were the steps that we followed, and we saw that there are certain bacteria which gives color with the gram stain, but there are some which will not give the color, which were not giving the color with the gram stain. So the one which were giving the color with the gram stain, they are the gram positive, right? And those who were not giving, they were the gram negative. So in the case of this gram positive and negative, the main difference was of cell wall. So in the gram positive bacteria, the cell wall was very thick. Teichoic acid was also there. In gram negative bacteria, that thick cell wall was not there. Along with that, in the cell wall, the outer part was having the lipopolysaccharides, right? So that lipopolysaccharide, what they did, students, right? That lipopolysaccharides, due to that lipopolysaccharide, what happened, beta? Yes, everybody please answer, right? Due to that polysaccharide, what happened? The lipopolysaccharide, we know that some porins were also there in that outer layer, and due to which, my dear students, due to which what happened, right? Due to which the stain comes out when we added the, yes, when we added what? When we added the decolorizer. So that's something that we covered in the last class. This is the diagram, right? And this is the diagram. So here you guys can clearly see this is the outer lipopolysaccharide. So here you people are having the porins. Here you people are having the porins. You guys can see that. Okay, so the stain will come out. The stain will come out. That is something we covered in the last class. Okay, now the next thing that we have to discuss is the cytoplasm and rest of the organelle. But before that, let's have the NCERT reading. Okay, so here you can see most prokaryotic cell, the bacterial cell, they have chemically complex cell envelope. And when it comes to the cell envelope, you have three-layered structure. Remember this thing, students? Three-layered structure. So in that three-layered structure, the outermost is glycocalyx, the middle one is cell wall, and the innermost is cell membrane. That's what we covered in our last class. Is that clear? So when your glycocalyx is tough, it is made up of polysaccharide, of course. Tough, it is thick, it is capsule. When it is jelly-like, then it is slime layer. Okay, when it is jelly-like, it is slime layer. Okay, so all the three layers, right? All the three layers together, they are working like what? They are working like a single protective unit. They are working like what? They are working like a single protective unit. Is that clear? Is that clear? Right, so gram negative and positive part is also given there, that on the basis of staining procedure developed by gram, those that take up the gram stain are gram positive and others are gram negative. Okay, so slime layer, capsule, all done, functions all done. Now this is the part that we are going to discuss. Okay, right, so take out your pen and paper and start writing the notes as well. Right, first understand it and then students just write down the notes. So we will start with the cytoplasm of the prokaryotic cell. From where are we going to start? We are going to start from the cytoplasm of the cell. Okay, okay, so here in the cytoplasm, what you people are going to see, that cytoplasm is granular. When I'm saying granular, means some like granules, right? Some granules like structure, they are present in the cytoplasm. So it is granular, semi-fluid matrix. What's that? It is granular, semi-fluid matrix that lacks cytoplasmic streaming. That lacks cytoplasmic streaming, also known as cyclosis. Yes, so that lacks cytoplasmic streaming, also known as cyclosis, and membrane-bound organelle. Yes, you heard me right, that here in this case, right, you people are not going to see membrane-bound organelles. Right, there will be no membrane-bound organelle in the prokaryotic cell. That is why it is the primitive cell. Right, that is why it is the primitive cell. That is why it is the cell which lack a well-defined nucleus. It is not having the membrane-bound structures, membrane-bound organelle. That is why we say that prokaryotic cell forms one envelope system. Okay, prokaryotic cell form one envelope system. Is that clear? Is that clear? So this is about the cytoplasm. Now the next thing that we have to understand here is mesosome. And mesosome is something very, very, very important. Right, mesosome is something very, very, very important. So students, if you remember the diagram from the last class, right, I made something like that, if you remember. Right, if you remember, I made something like this. You remember? Mesosome, right? Mesosome, they are what? They are the membranous extension. They are again the cell membranous extensions. They are again the finger-like structure. So whenever you read this thing, now finger-like structure, it simply means that it is increasing the surface area. And why is it so? Why is it so? Because in the case of prokaryotic cell, you people will see there is a structure, some finger-like structure is there that is going to increase the surface area. And here it is having the respiratory enzymes. What you people are going to see, you people are going to see the respiratory enzymes. What you people are going to see, you people are going to see the respiratory enzymes there, right? And moreover, this mesosome, it's not always like that. It will be in the form of these, finger-like structures only. It can form the vesicles there. It can form some tubules there, right? So anything is there. Like the structural variation is there. It can form some vesicles, it can form some tube-like structure, some lamellae it can also form. So it is having the respiratory enzyme, so it will help in the respiration there. So you relate it with the eukaryotic cell. In the eukaryotic cell, you people talk about mitochondria, yes or no? In the eukaryotic cell, you people talk about what? You people talk about the mitochondria. So what this mitochondria is, right? What this mitochondria is, this mitochondria is the powerhouse of the cell. Remember? Powerhouse of the cell. Aerobic respiration occur there. That is having the enzymes. We know it very well. Now in the prokaryotic cell, you don't have any membrane bound structure. Okay, you don't have any such organelle called mitochondria. So in the case of that prokaryotic cell, that function will be done by mesosome, because mesosomes are having the aerobic enzymes. Mesosomes are having the respiratory enzymes. So they will help in that respiration, number one. Plus, they help in the cell wall formation, DNA duplication, distribution of that daughter DNA to the distribution of that DNA to the daughter cells. So this is what we need to remember. This is what we need to write. And as I said, it is very important. So you please write it down that it is a special membranous structure. What is this mesosome? It's a special membranous structure. It is a special membranous structure. What's that? Special membranous structure which is formed by extension of, which is formed by extension of course, the extension of plasma membrane here, right? Extension of what? Extension of plasma membrane. Plasma membrane will extend and will form it. So these extensions are in the form of, these extensions are in the form of vesicles, tubules, and lamellae. These extensions are in the form of vesicles, tubules, and lamellae. Is that clear? Is that clear? Yes? Is that clear? Yes, everybody? So vesicles mean some, round structure like this, tube-like structure, some tubular structure. Lamellae means something very thin, right? Very thin lamella, kind of layer-like structure, right? A very thin layer kind of structure, right? So this is what the mesosome is. So now when you talk about the mesosome, so they help in, it helps in cell wall formation. You have to write it. It's a PYQ if students. So it helps in cell wall formation, DNA replication, distribution of that DNA to daughter cells. Distribution of that DNA to daughter cells. Is that clear? So it will help to distribute that DNA to daughter cells for secretion purpose. So they also play a role in secretion and have more surface area, or I would simply say that, and have respiratory enzymes, right? So how do they help? They help in cell wall formation, they help in DNA replication, distribution of that DNA to the daughter cells for the secretion purposes as well, and have respiratory enzymes, right? And have respiratory enzymes. Of course, that enzymes are going to help in respiration. That enzymes are going to help in respiration, isn't it? Isn't it? right? So finally, electricity. Oh. Okay, so finally, and have respiratory enzymes here. Is that clear? So this is the role of, or I would say the function of your, yes, this is the, once again, this is the function of what? This is the function of the mesosome here. Is that clear? This is the function of the mesosome here. Is that clear? Is that clear, everybody? Yes. So that's something you have to remember. So what is it? It is the special membranous structure, or I would say membranous extension. Right? Membranous extension. Am I clear? Am I clear? So the same thing is in the NCERT as well. So see, special membranous structure or the extension, it is mesosome, right? And formed by the extension of plasma membrane. So that is why in simple words I have mentioned it. It's a special membranous extension. Ambika. So it's a special membranous structure formed by the extension of plasma membrane. Formed by the extension of plasma membrane. Understood? Now there is one more thing that you have to remember. That is chromatophores. What's that? That is chromatophores. So students, do you know about chromatophores? Yes, anybody here in the class? Anybody here in the class? Any student from my batch? Here in the chat section right now? Yes, please. Chromatophores. So chroma word, it is associated with color. Chroma means color. So chromatophores are, these are some other membranous extensions. These are some other membranous extensions that are found in, yes, that are found in photosynthetic bacteria. Yes, please. So what are they? They are other membranous extensions that are found in photosynthetic bacteria, my dear students, and have pigments for photosynthesis. Are you getting my point? And have pigments for photosynthesis. So this is what we have, right? Chromatophores. What do we have here? Chromatophores. So chromatophores, they will contain pigment. This is another membranous extension. So obviously you will find it in photosynthetic bacteria. Is that clear? Is that clear? So can I say that in the case of cyanobacteria, we have chromatophores? Because we know that cyanobacteria, it is the blue-green algae, and this blue-green algae is responsible, right? This blue-green algae is responsible for what? This blue-green algae actually is the photosynthetic bacteria. Is that clear? So yes, it needs the pigments for the photosynthesis as well. So chromatophore is that another membranous extension that contains such pigment. Is that clear, beta? Is that clear? So it is the other membranous extension that contain what? That contain the pigments. Okay? Right? So next structure that we have to discuss here is ribosome. Ribosome. We don't have much here. In the eukaryotic cell, we have so many organelles. Here, you have this only, right? Here, you have this only. What is it? It is ribosome. So what is a ribosome, students? Firstly, remember this. It's a non-membrane bound organelle. What's that? It's a non-membrane bound organelle. It doesn't have any membrane. Plus, you call it as universal organelle. Why do we call it as the universal organelle? Because it is not just present in prokaryotic cell, but in eukaryotic cell. So this pigment is present in both, right? This pigment is present in both prokaryotic cell as well as eukaryotic cell. Prokaryotic cell as well as eukaryotic cell. In both the cases, you people are going to get it. What? Ribosome. Okay? Non-membrane bound organelle. So here, in prokaryotes, 70S ribosome. In prokaryotes, 70S ribosome is present in the cytoplasm. Right, beta? Which ribosome is present here in the prokaryotic cell? It is the 70S ribosome that is present in the prokaryotic cell. right beta? It is the 70S ribosome that is present in the prokaryotic cell. Now what is this S here? What is this S here? That's what we have to understand. Okay, fine, ma'am, got it. That ribosome is a non-membrane bound organelle. Ribosome is a universal organelle, right? So, but what exactly is this S? It is 70S, we got it, but what exactly is this S? Right, so this S stands for Swedberg constant. This S stands for Swedberg constant, and it is also the sedimentation coefficient. It is also the sedimentation coefficient. It's the sedimentation coefficient and it is the Swedberg constant. When you check the cell, right, or you study the cell, you go for something known as cell fractionation, right? You go for something that is known as what? Cell fractionation. So cell fractionation, this is the word that I'm using. You just listen to it, right? There is no need to write it, but yeah, you should pay attention here. So when I use the word cell fractionation, okay, cell fractionation, you are dividing the cell, basically, yeah, its component into different fragments. Yeah, I would say different. Basically, you are dividing something into different components. Yes, beta. So basically, when we are talking about this technique, that is cell fractionation, we are basically separating the different components, including the organelles, including the macromolecules, right? So you are separating it. What are you doing? What are you doing? You are separating it, right? And you preserve their individual factors. So basically, we are separating the organelles, the macromolecules, and we are going to preserve their individual factors as well. Now, what is going to happen in this cell fractionation, right? It is with the help of this technique, we separate the cellular components. Right, with the help of this technique, what are we going to do, students? We are going to separate the different cellular components. Okay, with which technique? I'll write the word here. Cell fractionation. I'm not saying that you have to include it in your notes, right? But yeah, try to understand the meaning. So fractionation means you are dividing something into different fragments. Okay, so you have to basically separate the cellular components here. Understood? 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 Yes, students, understood? Right? So here what we do, there are basically homogenization is there, right? And centrifugation is there. Homogenization is there. And then what else do you have? You have the centrifugation. You have what? You have the centrifugation. So what exactly is the meaning here? So when you are talking about homogenization, basically you are disrupting the cell wall and all. You are disrupting the outer covering. And in the centrifugation, of course, on the basis of size and density, you are separating the things. You are basically, first of all, let's suppose you took a tissue, right? You are going to form a mixture out of that. You are going to disrupt all that cell wall, outer coverings, and you are going to make a kind of I would say, if I have to use the appropriate word here, so I would say that you'll do what? You will disrupt their components so that everything can get mixed, and on the basis of size and density, you'll separate the components. Like you'll use the use these ultracentrifuges, and basically on the basis of size and density, you'll separate the components. Okay, you'll separate the components. They say, let's suppose you are doing it, right? There is a particular speed. Let's not enter into the depth of that. So you are getting the nuclear components first, and then later on other organelles like this. So of course, when you are doing this centrifugation, on the basis of size and density, things will settle. So it has to do this Swedberg constant or this sedimentation coefficient. It has to do something with this particular part, right? It has to do something with this particular part. So what's that? What's that? Let me tell you what's that. So here, this Swedberg, like 1s is equals to 10 raised to power minus 13 seconds, right? 1s is equals to 10 raised to power minus 13 seconds. So this Swedberg here, it is telling me about the time taken by a component or a particle, to form sediments, to settle down. In simple words, I'm explaining you this, that what is happening here, that when I'm saying S is, right? When I'm saying S, when I'm saying this S, when I'm saying the Swedberg, so it is telling me about the time taken by a particle to settle down or to form the sediments, right? Or to form the sediment. So time taken by a component or the particle to settle down or to form the sediments while the centrifugation. So when it comes to the S, right? When it comes to the S, so this S is the indirect measure. Again, it is written in NCERT, but in eukaryotic part, it is an indirect measure of what? Indirect measure of size and density, my dear students. It is not directly telling us about the density of a particular substance or the size of a particular substance. Rather, it is telling us about the time taken by that particular substance to settle down, right, during that centrifugation. Right, time taken by that particular substance to settle down during that centrifugation. That is the meaning here. Okay, that is the meaning here. Is that clear? That is the meaning here. This is your Swedberg here. Okay, this is your Swedberg here. Is that clear? Is that clear, everybody? Is that clear? This is your S here. So indirect measure of size and density, not the direct measure. So it is telling us about the time taken by a substance to settle down. So obviously, if something will be of large size, if it is having the compact structure, it will settle down quickly in comparison to the one which is comparatively smaller. Okay, so in that case, the one which is settling down very quickly, so it will have large Swedberg value. So Swedberg value is telling us about that, indirectly telling us about the size and density. That's what you have to remember. So when you are talking about the prokaryotes, right, when you people are talking about the prokaryotes, you have 70 S ribosome here, my dear students. You have 70 S ribosome here, and that 70 S ribosome is having two subunits, right? It is having 50 S as large subunit. Now you know the meaning of S here. And here it is the smaller subunit. So students, it's not like that, right? It's not like that you have to add it, that 50 plus 30 is 80. Of course not. 50S is the larger subunit, 30S is the smaller subunit. That's what you have to keep in your mind. And this ribosome is made up of nothing but the ribosomal RNAs and proteins, right? It is made up of the ribosomal RNA and proteins. So sometimes we also call this ribosome as ribonucleoprotein, because it is made up of ribosomal RNA and proteins. It is made up of ribosomal RNA and protein. So 50S is made up of different ribosomal RNA, 30S is made up of different ribosomal RNA. So both the subunits join only at the time of protein synthesis with the help of magnesium ion. With the help of what? With the help of Mg2 positive ion, magnesium ion. Is that clear? Is that clear? So let's suppose this is the larger subunit. Okay, this is the smaller subunit. So remember this, you will join them at the time of protein synthesis with the help of what? With the help of magnesium ion. And this is something important, right? This is something important. This is something you have to keep in your mind. Okay, so with the help of what? With the help of magnesium ion. Done. So now in prokaryotic cell, the another thing that you all see is that in prokaryotes, many ribosomes, right? Many ribosomes are attached. Many ribosomes are attached to a single mRNA chain. It is attached to a single mRNA chain and form polysome or polyribosome. So what am I trying to tell you here? Like imagine in the prokaryotic cell, this is the chain that you have. Which chain? This is the mRNA chain. What's that? It is the mRNA chain, right? mRNA chain. So here to this mRNA chain, single mRNA chain, so many ribosomes are attached, and you know, simultaneously, right students, simultaneously, like together, the protein, the transcription translation is going on. Together that transcription translation is going on. It is paired, like transcription and translation, you will discuss it in 12th. So those who are dropper, they understand it very well. Transcription means DNA will form mRNA. Translation means mRNA will form protein. So that's what we see here. That's what we discuss here, right? Right. So this type of structure where so many ribosomes are attached to single mRNA chain, we are calling it as polyribosome. And this is something you see in the prokaryotic cytoplasm. Up to this part clear? Yes, students. Up to this part all clear? Please tell me. And make sure you write the notes. Done? Yes, everybody, is it clear? So the next thing, right, next thing that we have to discuss is nucleoid, okay? Next thing that we have to discuss is nucleoid. Students, when you talk about the nucleoid, right, it is basically, it is also known as nucleoid, genophore, incipient nucleus. It is nucleoid, genophore, incipient nucleus. It is nucleoid, genophore, incipient nucleus. You also use the term prochromosome here. You also use the term naked nucleus or naked DNA here, right? This is also the term that you use. Now, what's the point here? Nucleoid, right? Here, nucleoid. Remember, it is devoid. Devoid means without histone proteins. Devoid of histone proteins, without histone proteins. Now let me elaborate. Let's take the example here. This is the prokaryotic cell that you have. Now this prokaryotic cell is having this nucleoid, right? We know it very well that prokaryotes, they have no well-defined nucleus, right? They don't have well-defined nucleus. So this double-stranded circular DNA. So this double-stranded circular DNA. What is it? Double-stranded circular DNA without histones is present in the cytoplasm and is not covered by nuclear membrane. Are you getting my point? So this is not even covered by nuclear membrane, right? This is not even covered by nuclear membrane, but overall the DNA is double-stranded, it is circular, right? And it is without histone proteins. About histone proteins, you discuss in the eukaryotic cell. Here you people are not going to see the, right? Here you people are not going to see the histone proteins. That's something you have to keep in your mind, okay? That's something you have to keep in your mind. Is that clear? Is that clear? Right? So this is the nucleoid, the genophore, right? Nucleoid, genophore, incipient nucleus, right? Prochromosome and naked DNA. Okay, so double-stranded circular DNA without histones, right? It is there in the cytoplasm, not covered by nuclear membrane. Moreover, remember this thing, students, that your prokaryotes, they are haploid basically, right? They are not diploid, they are haploid basically, right? They have that single set. And of course, if the DNA is circular, it will form one chromosome only. Understood? Understood? Sure. Now, the next point is that what is the use of these histones and why are we talking about the histones here? Students, in the case of eukaryotic cell, you talk about the DNA, right? We discuss the packaging of DNA. We discuss that this negatively charged DNA, it coils over the positively charged histones. Positively charged histones, and then it will form what? It will form the chromatin. Okay, then further it will pack, condense, it forms a chromosome. That's a general story. So that histones which are positively charged, right? Right, these histone proteins, what are they, students? They are positively charged, or I would say that they are the basic proteins. Positively charged and basic proteins, of course, it's same. Okay, so that's what we have here, right? That is the meaning of histones here. Now, here in the case of, right, in the case of prokaryotes, they are absent. Right, they are absent in prokaryotes. So in prokaryotes, what you people are going to see in prokaryotes, yes, students, in prokaryotes, you see some polyamines. There are some nucleoid associated proteins also, but you see polyamines. So these polyamines are also having positive charge. So they also help in forming the loops, right? Loops of that prokaryotic DNA. Is that clear? Yes, everybody. Is that clear? Done? So this is about the nucleoid. So this is an important point here, right? Do not miss this. This is important. Done. So other than this nucleoid, we need to discuss about one more thing that is known as plasmid. So do you know what plasmid is? Do you know what plasmid is? Yes, students? Do you know what plasmid is? Anybody? So plasmid is what? Plasmid is the extra chromosomal DNA. What is this plasmid? Plasmid is the extra chromosomal DNA, right? Of course, it is also double stranded. It is also circular and provide a unique phenotypic, provide the unique phenotypic characters, unique phenotypic characters. Yes, please. So it is the extra chromosomal DNA, which is double stranded again, which is circular again, and provide the unique phenotypic characters to bacteria. It will give unique phenotypic characters to bacteria. Now, what's the meaning of that? See, students, in some prokaryotic cells, right? In some prokaryotic cells. So when I'm saying some, so plasmid present. Now, what is this plasmid? Just now I told you, yes, please. Just now I told you that it is the extra chromosomal DNA. Just imagine it like this, right? It's a ringlet of DNA. It is the extra chromosomal DNA. This is your plasmid. Now, students, when you talk about this plasmid, of course, it's a DNA only. If it is a DNA, it is going to have the genes, yes or no? Yes or no? Please tell me, yes or no? If plasmid is the DNA, definitely it is going to have the genes. It is going to have the genes. And we know that a gene is going to express itself by making proteins, right? By making proteins. And that are the proteins, right? That are the proteins which are going to give a particular type of physical appearance. Like we got our genes from our parents, that genes are making proteins, that proteins are expressing, and we are getting a particular character which resembles with our parents as well, right? Not completely, but yeah, similarities are there. So of course, if here also we have the DNA, so that if it is the DNA, it is going to have the gene, it will express itself. So the genes which are present on plasmid, right? Because not every bacteria is having the plasmid. So genes which are present on plasmid, my dear students, the genes which are present on plasmid, my dear student, in that case, what you people are going to see, right? In that case, what you people are going to see, you will see that these genes will make protein that will give a unique phenotypic character to this bacteria. Like might be you can have the gene for antibiotic resistance here, right? We can have the gene for antibiotic resistance here, right? Let's suppose because of the gene on the plasmid, the bacteria is not getting killed with the antibiotic A. That can be the possibility. Okay, maybe it is having some drought resistant genes, right? Drought resistance genes. So this type of things can be there. Okay, this type of things can be there. So this is the plasmid, and this plasmid can replicate. It is a self-replicating, right? Self-replicating circular DNA. It doesn't depend upon the depends upon the nucleoid for its division. It's self-replicating, right? It's self-replicating. Understood? Understood? Done? Sure? So what is this plasmid? This plasmid is the self-replicating circular DNA, provide unique phenotypic characters to the bacteria. So we discuss about the plasmids in the biotech, class 12. In class 12 in biotech, you will see that how, these plasmids are used as the gene taxi or the vector. They'll help us to transfer the genes from one cell to another. Understood? Okay. So the next thing that we have to discuss here is flagella, my dear students. What we have to discuss? Flagella. So if it is flagella, definitely, like see, if bacteria is motile, what is it? It is motile. It contains flagella. It contains flagella present. Right? What is going to happen if the bacteria is motile? What's going to happen? The flagella is present, right, students? So this flagella can show, like no doubt, in the bacteria you have the flagella. So that flagella can have, different type of arrangement can be there. right? So in the case of prokaryotes, this is what you people are going to see, right? In the case of prokaryote, this is what you people are going to see, that when it comes to the flagella, that in the bacterial cells which is having flagella, so they show the different range and the different, they will show the different arrangement of that flagella on the bacterial cell. It's different range and it's a different arrangement on the bacterial cell. Like for an example, if I have to give you the example, some bacteria have no flagella, right? No flagella. So I can call it as atrichous. It is immotile. Yes, you can say so. It is atrichous. Now let's suppose if there is one flagella, monotrichous only. There can be many flagellas at the head region only. There can be, many flagella which are covering, right? Just like the fimbriae or the cilia, which is there in cilia is there in eukaryotic cell, na? So that's what you people are going to see. Are you getting my point? That's what you're going to see. So yeah, there is a different arrangement. Is that clear? The arrangement is going to be different. Understood? The arrangement is going to be different. Is that clear? Sure? Yes? So NCRT, in the NCRT it is written that if bacteria is motile, flagella present, or how in the NCRT it is mentioned that a thin filamentous extension coming out from cell surface, yes students, is called flagella. That is the line of NCRT. Now I have to elaborate it. I have to discuss it with you all. Right? I have to elaborate it. I have to discuss it with you all. So that is coming out from the cell surface to the outside. Right? That is coming out from the cell surface to the outside. Or here, I have used the word cell surface, that there is a thin filamentous extension coming out from the cell surface. If you read it from the NCRT, in the NCRT, they have mentioned cell wall, that it is coming out from the cell wall. Now here I need to classify, I need to clarify two or three things. Wait, I'll show you. Like here, please read this line and kindly pay attention. You can say bacteria may be motile or non-motile. So if it is motile, like I said, they have thin filamentous extension coming from their cell wall called flagella. What are they saying? Thin filamentous extension. What are they saying? What is the language of NCRT saying? Thin filamentous extension. Actually, students, flagella, this flagella in prokaryotes, right? This flagella in prokaryotes, I'll ask you to write as well, but let's understand it first. So in prokaryotes, flagella is made up of flagellin protein. Flagella is made up of flagellin protein. Which protein is making flagella, people? Flagella is made up of flagellin protein. It is made up of what? Flagellin protein, number one. Now, what's the point here? This flagella is having three parts here. How many parts? Three parts. Like you will have basal body, which is also known as blepharoplast. You will have hook and you will have filament. Basal body, hook and filament will be there. Basal body, hook and filament will be there. So what will you see here? Like let's suppose this is the cell, bacterial cell. You will have a basal body, you'll have a hook-like structure, and this portion will come out. I'll show you a proper diagram as well, but you understand this first. They say this is the structure. I'm calling it as basal body. Then there is a hook, and then a thin thread-like structure. Filament is coming out, and that filament is the longest portion of your flagella. Now, when you look at it, now here in the NCRT, they are clearly mentioning cell wall. That from cell wall, this extension is coming out, right? So bacteria show a range in number and arrangement of flagella, right? Bacteria show a range in number and arrangement of flagella. It is written here. So now this is something that I was elaborating here, and this is something I need to highlight here as well. So clearly I have mentioned if bacteria is motile, a thin filamentous extension is coming out from cell wall. As per NCRT, from cell wall is called flagella, and bacteria shows a range in number and arrangement of flagella. Bacteria show a range in number and arrangement of flagella. This bacteria is going to show arrange in number and arrangement of flagella. Now, next slide, you have to mention it in this way only, that flagella is having how many parts? 3 parts are there: hook, basal body, and filament. Right? Hook, basal body, and filament. So this filament is the longest portion. This filament is the longest portion. But now, when I look at the diagram, see this. That's what I'm talking about. I'm talking about the flagella here. Okay, I'm talking about the flagella here. I told you it is showing the different range. It is showing the different arrangement, right? This is what I'm talking about. Okay, this is what I'm talking about. You can see this diagram. Now, when you look at the structure of the flagella, right, basal body. See, firstly, look at this part. This is the plasma membrane. What's that? This is the plasma membrane. Now, this is the plasma membrane. This is actually a gram-negative bacteria. This is the image of a gram-negative bacteria. How can we say that? It is having the plasma membrane. It is having the thin peptidoglycan, plus it is having one more outer membrane. So when you look at this diagram, right, you see that the basal body is there in the plasma membrane first, right? This is also the portion of basal body only. This is also the portion of basal body only, right? This part, this basal body, firstly, it is there in the plasma membrane. Then you can see it is there in the cell wall as well. Then we have the structure known as hook beta. And then this is the thin filament, right? Which is made up of which protein? Which is made up of flagellin protein. Which is made up of which protein, students? Which is made up of flagellin protein, isn't it? Which is made up of which protein? Flagellin protein. Am I clear here? Am I clear? Is it understood? Now, we are saying that flagella is coming out from the cell wall, right? Is it actually coming out from the cell wall, right? Cell membrane is also there, like that is also containing the part of the basal body. So basically, I want to tell you that if you read that line carefully, right? If you read that line carefully, which one? The one that we just read from the NCERT, right? This one, that bacteria show a range in number and arrangement of flagella, and this flagellum is having 3 parts, and the filament is the longest portion, which is extending from cell surface to the outside. This point is absolutely correct. That filament, that thin structure is coming out from the cell surface and then it is coming out to the outside. But here, the point which is written here, where is it? That yes, that bacteria show a range in this point, that if motile, they have thin filamentous extension coming from their cell wall called flagella. So if somebody is asking you that where that basal body is embedded, that is also there in the cell membrane, not just in the cell wall only. But now here in this line, they are saying that filamentous extension from cell wall is coming out. So basically we can take it in this way that, after that basal body, hook and then filament is there. So that is somewhere coming out from the cell wall. So in this way you can take it. But if somebody is asking you that where that, right, where that? Yes, where are the where is the basal body? So then you should know. Okay, understood? Yes. Okay. So filament, longest portion, so extend from cell surface to the, right? So you can write it here that it extends from cell surface to outside. It extends from cell surface to outside. Okay, so in prokaryotes the flagella is, so in prokaryotes flagella is very simple. In prokaryotes flagella is very simple and made up of flagellin protein. Made up of flagellin protein. Which protein is making it, students? It is the flagellin protein. Which protein is making it, my dear students? It is the flagellin protein that is making it. Understood? That is making it. Sure? Okay? Done? Done? So next thing that we have to discuss is pili and fimbriae. Okay, next thing that we have to discuss is pili and fimbriae and inclusion body, and then of course the prokaryotic cell part will be over. So write down. So next part, pili and fimbriae. So students, here in the prokaryotic cell, they don't just have flagella. You will also see that in some bacterial cells, some other fiber-like structures, they are sprouting out of cell, and we call them as fimbriae. What do we call them? We call them as fimbriae. So how do they help? They help in attachment. They help in attachment. Attachment to substrate or the surface or host cell. It's not something that is playing a role in the motility, not like that, right? Not like that. Neither pili nor fimbriae play a role in motility, they just help in the attachment. So this is about the fimbriae. So you can write down that besides you understand, in addition to, right? What is the meaning of besides here, beta? What is the meaning of besides here? So beside here simply means that in addition to, so there are some other surface structures. So beside flagella, pili and fimbriae are some other surface structures. Okay, there are some other surface structures, but what is the point here? What is the point here? Structures that do not play role in motility. Okay, so it is not playing any role in movement. That's what you have to keep in your mind, right? It is playing no role in movement, but rather they have the different functions, like as I said, fimbriae. Yes, so like I said, the fimbriae, they help in what? They help in what? They help in the attachment, right? They help in the attachment. So they are small bristle-like fibers, right? What are they? They are small bristle-like fibers sprouting out of cell surface. Okay, so it helps bacteria to get attached to substratum or host cell. Are you getting my point? Substratum or host cell. So I'll give you the example here. I'll give you the example of a bacteria here, that is Neisseria gonorrhea. Actually, students, this particular bacteria, it causes the reproductive tract infection gonorrhea, right? Neisseria gonorrhea it is. So this particular, right? What is happening here? That this particular bacteria, it is responsible for causing, right? It is responsible for causing what? The gonorrhea disease, the reproductive tract infection. So this enters in host cell with the help of fimbriae. With the help of fimbriae. Okay, that's the one thing. Noted, Biche? That's the one thing. Next point, pili. What are they? First of all, it is made up of pilin protein, right? So what are they? They are the elongated tubular structure, tube-like structure. So it is the elongated tubular structure made up of pilin protein. Okay, so let's understand this part. What exactly is it going to do? So students, so bacteria having F plasmid. So it's not like that all the, right? It's not like that all the cells are going to make it, right? All the bacterial cells are going to make a structure like this. No, It is not the case, right? It is not the case that all the bacteria will form the structure like this. So bacteria having F plasmid. Here F stands for fertility. Here F stands for fertility. So bacteria having F plasmid form such tube-like structures. Mainly we see them in the case of, gram-negative bacteria, right? Such tube-like structure. Like for an example, let's suppose this is the bacteria. This bacteria is having one more plasmid. You call this plasmid as F plasmid. What is the name of this plasmid? F plasmid. It is even considered as donor, my dear students. It is even considered as male here. Male here. So now this bacteria, right? Now this particular bacteria, because it is having such plasmid, it forms a tube-like structure. Are you getting my point? It forms a tube-like structure, something like that, some tube-like structure. So this is known as pilus. It is also known as sex pilus because it helps in genetic recombination. Let me discuss that with you all. Let's suppose this is another bacteria. Let's suppose this is another bacteria. It is F minus. Why? Because it lacks F plasmid. It lacks F plasmid. Okay, because it lacks F plasmid. Right? It is even considered as female. So what's going to happen now? This bacteria, with the help of this tube-like structure, will get attached to, right? This bacteria, with the help of this tube-like structure, get attached to this particular bacteria here. Now this plasmid will replicate, this will divide, and finally the plasmid will be passed here, right, to this cell as well. So by the end, you people will see two bacterial cells. Now they both will have the F plasmid. Now they both will have the F plasmid. So that's how this pilus will help here. That's how this conjugation tube will help here. And this is something you need to keep in your mind. Is that clear? This is something you need to keep in your mind. Is that clear? So mostly in gram-negative bacteria, you'll see the structure like that. And this phenomena that I have discussed here is conjugation. We'll discuss about it in biological classification. So this is the conjugation, right? This is the conjugation. It is considered as a primitive sexual reproduction in bacteria. Though it's not a true sexual reproduction, right? Though it's not a true sexual reproduction, no gamete formation is there, right? It's a kind of genetic recombination because one cell is passing this plasmid to the another. Understood? To the another, right? No, it is not the tubulin protein. It is the pyelin protein. It's the pyelin protein that is making it. Okay, so now the last topic of this prokaryotic cell that we have to discuss is inclusion bodies. What we have to discuss? Inclusion bodies, right? So we got so many questions from this part, right? Even in 2020 question was there. If I'm not wrong, 2022 is also having, in the 2022 NEET 2022 examination also, the question was there from the inclusion body. In 2020 it was there for sure, right? Rest it was there in 2021 or 2. I need to check the year there. So inclusion bodies, what are they? First of all, they are non-membrane bound structures. They are not membrane bound. They are not, I would say, not covered by any membrane. This is something very important that you need to keep in your mind, that they are not covered by any membrane. That are not covered by any membrane, right? So non-membrane bound, of course. And what exactly it is? It is actually the reserve food material. It is actually what? It is actually the reserve food material stored in cytoplasm. It shows what? It shows the reserve food material which is stored there in the cytoplasm. Okay, which is stored there in the cytoplasm. So reserve food material is stored in cytoplasm as inclusion bodies in prokaryotes. Okay, in prokaryotes. Am I clear? So you have many examples, but the important point is this. This is not covered with any membrane. Okay, this is not covered with any membrane. Okay, so here you have the example like glycogen granules, phosphate granules. Yes, you have glycogen granules, phosphate granules, cyanophycean granules are also there. Am I clear? Am I clear? Yes? Yes? So now other than that, students, gas vacuoles, right? Gas vacuoles are also found in some blue-green, green and purple green sulfur bacteria. So these gas vacuole, as the name is right, so these gas vacuoles, as the name is suggesting, gas vacuole, so of course it will have some, you know, metabolic gases, isn't it? Right, so firstly blue, green, purple and green and purple bacteria. Of course it is the kind of photosynthetic bacteria here. So this gas vacuole, okay, it is membrane bound actually, right? It is membrane bound. In the prokaryotes, if you will see the gas vacuole, it is membrane bound. No beta, inclusion bodies are only there in the prokaryotes, right? So gas vacuole membrane bound, but that membrane is, okay, but that membrane is non-unit membrane. Like it is not the unit membrane, it is the non-unit membrane, right? It is having some proteins. Some proteins are going to make it. Yeah, non-lipid unit membrane it is. Okay? Okay, so this is about the gas vacuole. This is about the gas vacuole. Understood? Understood? So firstly, write down the points. Yes? Right? So basically, it's not. So I'm saying it is membrane bound. So that membrane, as I said, it's not a true membrane. Like we discussed phospholipid bilayer or something. So it's membrane bound, but that membrane is a non-unit lipid membrane, or I would say that membrane is not a true membrane. It's the non-unit lipid membrane, or you can say it is basically a kind of protein membrane. It is something made up of protein. Like because here in the gas vacuole, of course, they'll have some gases, right, formed by the aggregation. So of course, a kind of protein will form a covering around it. That membrane we are talking here, not that proper phospholipid membrane, right, and all. This is not something that you are going to see here. Right? So this is all about the prokaryotic cell. Okay, you even if you read it from the NCERT, you'll find it that we have discussed each and every point, including your ribosome, inclusion body, everything. So the next topic that we have to discuss is eukaryotic cell, and of course that we will discuss in the next class. Okay, so as you people said that you need a plan for this series, and I told you last time also that I will start taking your classes regularly here in the series. Right, I'll start taking your classes regularly, right? Even if it is for 1 hour, 1.5 hour, 2 hours, depends upon the need, right? We will have these particular classes regularly. Don't worry about that. But you keep studying and your syllabus will be completed. You know that this channel is known for its marathon, right? We'll come for a day, we'll complete the chapter, and then slowly you can revise it. So there is nothing to worry about. So you just keep on studying. And guys, as I said that from 1st of August, I'll be starting anatomy of flowering plants, right? I'll be starting anatomy of flowering plants in my Phoenix 3.0 batch, right? In 3.0 batch. If you ask me about the syllabus completion, I am considering it as 15th of Jan. And it's not like that if teacher is saying 15th of Jan, they'll stretch it till 31st. No. No, I have checked everything. All the possible calculations are made, and on the basis of that, I'm telling you the syllabus, biology syllabus, will be completed by 15th of Jan. And why is it so? Why not December? Because I have seen that later on students, they don't study, they don't revise, they don't practice the question. So in the class itself, right? In the class itself, we will have the practice session. Like today, we are about to finish our chapter biomolecule, and after that there will be the MCQ session, their MCQ practice session. There will be the DPP session. Like in the today's session only, we'll discuss everything there, right? We will discuss everything there. So it's like that. It's like that. So that's what you have to do, right? So I am starting this chapter, and after that plant physiology will be there. So without any delay, you join the batch. Now if you are having the concern that, ma'am, what about the chapters that you have already completed, right? Okay, I will, for the cell, for cell cycle, and for biomolecule, for this complete unit, I'll start taking the classes, right? I'll start taking the live classes as well, that too in the month of August only, right? Right, I'll keep a different slot and we'll do the same. So without any delay, be the part of the batch, and you know how to enroll in the batch. You can go to our channel, you can go to the description box of this particular video, you can click on the link there, and you can enroll yourself. Okay, so that's it, that's all. So do let me know how much you like the session. So take care, do revise it. If there is any doubt, right, you can mention that in the comment section. So thank